Thank you. So today I'll be talking about the genome editing capabilities of Demeetra and unveiling some advantages of Cas-CLOVER over your standard CRISPR-Cas9. So a little bit about Demeetra. We are located in Lexington, Kentucky. So we're known for bourbon, horses, UK basketball, maybe some gene editing here and there. But we were founded in 2019, and 80% of us are hands-on research. So we're really focused on optimizing these gene editing technologies and then transferring those for commercial applications. And we provide innovative licensing models with freedom to operate for cell line development, bioprocessing, agriculture, and synthetic biology. So Demeetra's core technologies are the harbor-in transposase transposon system. So this is very similar to your super piggyback transposase, where it would recognize that inverted terminal repeat and then excise that cargo from the transposon and then insert that into the genome at highly expressed sites. These are typically open chromatin and non-coding regions, and these are found to be very stable. The great thing about harbor-in transposase is that it's able to do large cargo. So we can do 175 kb, and there's been reports of 200 kb as well. Our second technology is Cas-CLOVER. It's a highly efficient targeted dimeric nuclease. So this utilizes an inactivated Cas9 molecule that's strictly there for DNA recognition. And then this is fused to a proprietary CLO51 endonuclease that is an obligate homodimer. So it needs its partner to actually do the cutting. And this really leads to lower off targets as well as lower translocation events that you would typically see with CRISPR-Cas9. It's very easy to use. The guide RNA is a plug-and-play infrastructure. So you can use the same softwares that you would use to find guides for Cas-CLOVER, and we even have some search tools that was developed by our scientists that help you go through there. And we're seeing large deletions with Cas-CLOVER because CLO51 is a restriction endonuclease, so it cuts like one too. So this actually leaves 4 base pair overhangs, and this can lead to higher knock-in efficiencies. Okay, so Cas-CLOVER is a fusion protein, and we often know that these are dynamic and difficult to express and purify. Fortunately for Demeetra, we have scientists that were able to produce the recombinant Cas-CLOVER protein, and this is used a lot to do in vitro assays for cutting mechanisms and kinetics. So this is a typical amplicon that we would design a left guide, if I can get this pointer, a left guide and a right guide for Cas-CLOVER and a right guide for Cas9. So we would incubate the protein with the guide RNAs, and then you would look at the cut products down here, and you can PCR them and actually find out how they're cutting. So this is showing that Cas-CLOVER is cutting right in between the left guide and the right guide with a 4 base overhang. And then Cas9, your very typical blunt cutting mechanism right next to the PAM site. So we're seeing improved indel efficiencies in CHO cells compared to Cas9. So this is using mRNA for Cas-CLOVER and Cas9. This is targeting a very popular gene for mammalian cell line development, glutamine synthetase. We're seeing 80%, 75 to 80% cutting efficiencies using a T7 endonuclease assay compared to Cas9's around 60%. And this is using a variety of different guide RNA scaffolds. So you can get guide RNAs from IDT or Synthago, and they have different tracer backbones, and they work very well for Cas-CLOVER as well. We're also using the recombinant protein that we previously just made, and this is working very well in compared to CRISPR-Cas9. Using the left or the right guide for Cas9, you're seeing pretty comparable efficiencies with our Cas-CLOVER protein as well. Now, not only is Cas-CLOVER very efficient at knocking out genes, we're seeing it has an enhanced targeted knock-in efficiency as well. So we targeted, we used GFP and puromycin, and that knock-in was around 7 kb in size. And we used HEC293 to compare the efficiencies of the knock-ins between Cas-CLOVER and CRISPR-Cas9. So right after post-transfection, we were around 10% with Cas-CLOVER. However, we were full fourfold lower with CRISPR-Cas9, about 2.5%. Now, when you do that puromycin selection, you can enhance that pool of edited cells to around 70% with Cas-CLOVER and only 35% with CRISPR-Cas9. So a little bit of summary about Cas-CLOVER compared to CRISPR-Cas9. You see nearly 0% off-target and translocations with Cas-CLOVER. However, these are kind of significant with CRISPR-Cas9. The activity is very comparable for Cas-CLOVER to Cas9 for knockouts, but we're seeing a greater efficiency of knock-ins compared to Cas9. We see very large indels with Cas-CLOVER. So Cas9, typically you get 1 to 6 base pairs if you're using a single guide. With Cas-CLOVER, you're seeing anywhere from 12 base pairs to 40 base pairs. Even if you have to have the left and the right guide, we're still frequently able to find guide pairs to target any gene of interest. I think the most important thing is our commercial freedom to operate. We just do a simple single license without any downstream royalties and everything that you have to do with CRISPR-Cas9. So we're using Cas-CLOVER to develop cell line development platforms. And obviously glutamine synthetase is still the industry benchmark for high titer suspension CHO cells. And that can be for expressing very difficult to express proteins. So we have very good editing for the GS gene in CHO cells. We see very nice deletions in our clones that we isolate. So here's the typical deletions that you're starting to see with Cas-CLOVER. And then we're also able to identify a great phenotype as well. So in our cell line development platforms, we are identifying clones that have greater sensitivity to glutamine, but we're also looking at important growth parameters like doubling times and everything like that. We're also developing, right now we're almost out with a GS foot 8 double knockout. So this would be to make afucosylated antibodies that have greater ADCC. With one guide pair at a particular exon, we're seeing about 80% cutting efficiencies with Cas-CLOVER, whereas Cas9 we see around 60%. We always like to compare it to Cas9 just to see how we're doing. We're identifying those unique clones. We're able to identify those nice deletions for each clone. And then looking at the phenotypes, we're doing LCA staining, which is staining the fucosylated N glycans on the surface. So in your normal CHO GS that has the wild type foot 8, you see nice green staining on the surface for those glycans, whereas in our top clone, you're not seeing any of that staining. Same thing for IF of the foot 8 protein. Very nice green and merged image here with a CHO GS wild type foot 8. And then you lack that GFP or FITC staining with our top clone. So our Harbor N transposase comes as a kit for the first time. So it has the same benefits as other license restricted transposases, but it's license free. So it comes as an optimized mRNA, and this is going to give you pretty high copies. So what we're seeing with the mRNA in CHO cells, you're seeing anywhere from 5 to 20 copies put into the genome. There's no IP restrictions and there's no license required. And you can use this for any field of use. It could be cell line development, cell and gene therapy, you name it. And we usually supply this kit with multiple transposons. So if you were to use the CHO GS cells, you could use a transposon that has your glutamine selection, and there's multiple cloning sites to pop in your genes of interest. So utilizing those clean-cut GS CHO cells and the Harbor N transposase, we were able to make stable trastuzumab-expressing cells. And when we isolate certain cells or certain clones, we're able to get antibody titers around 5.5 grams and an unoptimized 24 deep well plate with a viable cell density is only about 9.5 times 10 to the 6 per mil. So that's not very high. And we're seeing very high QPs for this, around 110. So these are quite the highest ones that we've seen throughout the literature as well. We take this a little bit further and collaborate with a CDMO. They're able to take this particular clone and put it in an amber system and get around 8.5 grams per liter. So this is kind of like hot off the press. We've pretty much made this slide like 2 days ago, but we're working on expressing and making this purified protein. So this is a very difficult protein to make. However, we've came up with a really nice system to actually produce and express this protein with specific advantages in mind. So it would work in many cell types. It would be a short-lived activity, reducing genomic instability. It can reduce off-target integrations. It can reduce the risk of plasmid integration, and it can lower the risk of insertional mutagenesis, which it would be safer for cell and gene therapy. So an in vivo EMSA assay, we did this with the purified harbor end protein with a transposon. So you can see the major species of the transposon labeled in 1. So that would be the whole plasmid just ran through the gel without harbor end. But then if you complex the harbor end protein with the transposon, you see a nice shift up in the gel where that complex cannot run through the gel. The really unique thing that caught our eye is this band right here. And this band is actually this backbone. It's getting released. So you're actually seeing the activity of the harbor end protein, grabbing it, excising it out of the transposon and releasing that backbone. So this could be a really unique system for cell and gene therapy or anything of interest. And when you treat this with proteinase K, that whole complex, you degrade the protein, and now you release all of your major species of your DNA, which was really exciting to see. So then as a proof of concept, we used the harbor end protein with T cells, and we actually put in a GFP, and there was 10% post-transfection of GFP positive cells. And then after sorting, we were able to get it to around 93% of GFP positive cells. And then we actually went in and used a variety of sequencing methods to identify which sites had transposition and the cargo of interest. And this is following a very typical piggyback-like transposase profile where a lot of it's non-coding in intronic regions. So lastly, I just want to conclude, we have a very comprehensive freedom to operate cell line development package. We have a lot of different licenses for a lot of different parts for this, but this is bundled nicely for clients with no royalties for that. We used that Cas-CLOVER to make the clean-cut GS CHO cell lines, and we have freedom to operate there with a license from ECAC. And obviously, we have Harbor In as a commercial reagent. So with that, you can please visit us at booth 806. I think we're doing a bourbon raffle too, for those that are interested. And then I believe a talented research scientist, Thomas, is doing a poster in the afternoon session. Thank you.
We have time for one quick question. I saw it right here.
Yeah, thank you for this really great presentation. I have a question regarding the harboring transposase. Apart from the capacity to embark large cargoes, what are the main differentiators against piggyback systems?
It works very similar to the piggyback system. We have certain patents pending for the production of this optimized mRNA and protein format. When we're comparing it to other available transposases out there that are piggyback-like, the vectors that they're using are very unoptimized for mRNA production. So then that correlates downstream to even worse results. So our products that we're selling is just a commercial reagent. They're just working a lot better. And they're still able to do that very large cargo and everything like that.
Okay, let's go ahead and thank the speaker one more time.